All right, I think we are ready for the webinar today. Please let me know in the chat section if you can hear me well, if you can see me, as well as if you can see my presentation on the screen. So put in one or yes in the chat section so that I can get the green light that we are ready to begin with today's exciting session. And that would be my confirmation that I'm ready. We are ready to go. All right. So we have got the confirmation. Thank you so much for letting me know. And once again, I welcome you all to today's webinar and to the to another meeting of the Psionics Student Club for Bioinformatics enthusiasts, bioinformatics lovers, students, faculties, researchers all across the globe. It's my immense pleasure to welcome you all today. And before we move on, let me introduce you to myself. I am Subrajit Barua, and I am the president of the Psionics Student Club, as well as I'm also the scientific outreach coordinator at Pine Biotech. The vice president of this club is Ms. Ruth Vivaja, who unfortunately isn't uh, available today because of her exams, but she is also an active member of the student club. She has been handling the club and working behind the scenes for a long time. So it's my pleasure to have her as my teammate as well. So let me briefly introduce you to Omics and the Psiomics Student Club. Well, we are not only a club, but we are also a community. We are a family of more than 1,000 students from all across the globe, from different countries, different areas. We also have a huge community of experts from who are expertised in different backgrounds, and they always help us, assist us, train us to become the better version of ourselves in the domain of bioinformatics, computational biology, and biology in general. We are also proud of our very strong alumni network who have been a part of the Psiomics Club for a long time. They have trained themselves in bioinformatics, they have learned the skills, and now they are making an impact all around the world in different areas and domains. And we are really proud of that rich network of alumni in our club. So, what is the vision of Psiomics, the Bioinformatics Club? Well, at Psiomics, we are really interested in different areas or applications of bioinformatics and computational biology. To name a few, we are interested in infectious diseases, microbes and microbiome, precision oncology, plant science, neuroscience, and even the very recent and exciting field of astrobiology or space omics. So, the club Psiomics is focused and driven towards clearing concepts of bioinformatics to help one another gain insights on how to use a bioinformatics tool to learn to apply these techniques in research in day-to-day life. We have brainstorming sessions to help one another grow and enhance in life. We also have sessions on coding skills and also on data science like we are having today. At Psiomics, in the past, we have also had some sessions which were dedicated to help the students get better in their academics, to apply abroad, study abroad programs, how to refine your CV, how to refine your curriculum vitae and resume, and how to crack an interview are some of the other non-conventional topics which we also cover at Psiomics. So if you feel that this is something which you are interested in and you would like to be a part of us, please consider joining us and staying with us. You are always welcome. Moving forward, you will hear the word OmicsLogic a lot, so I think it's mandatory for me to introduce what OmicsLogic is. Well, OmicsLogic is a community or an initiative which is aimed at creating and providing good quality training in bioinformatics, which enables the trainees, the ones who get the training, to be enabled to do independent research guided by mentors and peer examples. OmicsLogic is also focused towards developing a growing community such as this one, Psiomics, with the shared passion for data-driven research and appreciation of citizen science research as well. And finally, we also enable students, clinicians, and faculties from all backgrounds to develop new techniques, new tools, and algorithms which can be applied in life science research moving forward. Well, now I would like to briefly introduce you to the upcoming Omics Research Symposium that is going to happen on March 31st and April 1st. This is an international conference which is an exciting opportunity for all of you to be a part of. This particular symposium is held biannually, that is twice a year, which brings together the huge community that OmicsLogic has produced, which includes students, faculties, industrial experts, together on the same platform to discuss the latest trends, the latest advancements, the cutting edge research, their outcomes, as well as to discuss what are the various opportunities, what are the areas of concerns, and how to improve. Students can really gain insights about what industries are looking for in a student, what is it that takes a student to get placed, to get their dream job, and a lot of more interesting information. This time at the Omics Research Symposium, we have experts from the academia from leading universities such as Amity University, Georgetown University, and many other top universities of the world. We have industrial partners and speakers from organizations such as Google, NASA Ames Research Centre, Blue Marble Space, FABA Academy, Pistoia Alliance, Novo Nordisk, and many more. And also, we have experts from our network partners, which include LBRN, NEPI, H3A Bionet, and many more. So here I would briefly want to talk about what is it that the universities are looking for. Well, they are looking for involving capable students in exciting and cutting edge research. They're looking for placements for students. That's one of the end goal of universities, to enable their students get placed in reputed organizations, and also to perform translational research, that is a research from lab to the market. That is something which industries, the universities are looking for. What are the industries or the companies looking for? Well, they're looking for students to provide internship, co-ops, and trainings. They are also looking for potential employees, both young career opportunities as well as experienced employees are something which industries are constantly looking forward to. And also, these industries have a lot of outreach programs, a lot of training resources that they have made. So they want to get those people who can benefit from these resources. So, how can the Omics Research Symposium help all the three parties? Well, this is a platform, as I said, that brings together experts from both industries and academia on a common platform. We also have a large audience of students from all across the globe with diverse skill sets who will be a part of this symposium. And this symposium brings an opportunity for networking and getting to know each other better, to discuss various opportunities and to proceed ahead in life. So, what's unique about the students who are a part of OmicsLogic, Psionics, and who are also going to be a part of the Omics Research Symposium? Well, these students belong to great universities from all across the globe. And these universities are the universities that are partners to us. We are in constant touch with them, and we know the training standards, the training modules. Our students have a fine understanding of bioinformatics tools and concepts that they have gained by going through the various course materials available on the OmicsLogic platform that you can also access at your end to get trained in bioinformatics. And our students also know how to apply these techniques and tools, and that can be seen when they present their poster presentations. More on poster presentation in just a while. So here's a sneak peek into some of the great speakers lined up for the Omics Research Symposium 2022. You can find speakers from different backgrounds with different levels of expertise, all connected through bioinformatics and big data science together on this common platform. So if you find this interesting, do check it out and do make sure to register for the symposium. Now, apart from listening to these experts' talks and various cutting edge research and advancements, there's also another component of the symposium, which is a poster presentation competition. This is your opportunity for the students and researchers to showcase your projects, your experiences, just like these projects, as you can see on the screen, which were the ones which won the prizes, the cash prizes last time in the previous symposium. So here you can see examples of posters made by high school students, by undergraduates who not only learned about bioinformatics, got trained in bioinformatics, and also understood how to apply these techniques in solving some research problems, which is a real world problems, how to deal with data, how to analyze them, how to understand the results and present it as a poster. So that is the entire idea of organizing these webinars, organizing all the programs and modules, and finally to showcase all of these exciting results in the symposium. This time we have four categories in which you can submit your posters, namely Omics and Bioinformatics, Artificial Intelligence, Machine Learning and Data Science, Astrobiology and Space Omics, and Computational Biology for Biotech and Pharmaceuticals. The best posters will walk away with cash prizes segregated into three different categories, namely Best Poster Presentation, Best Citizen Science Project, and Young Sheldon Award for High School Researchers. So, you must be wondering what's the procedure of doing such projects? How is the best way to overcome challenges? Maybe you are working on something, but you are facing some issues with data handling or applying some tools and techniques. What is it that the judges of these poster presentations would be looking for from these posters? Well, that is why we have with us Mr. Darko Medin today, who is a data scientist and an artificial intelligence and statistics expert, who is also the judge of the poster presentation competition of the symposium. So today, Mr. Darko Medin will be talking about bioinformatics and data science challenges and what are the ways to address them and how you can overcome these challenges, complete your project and present it in this poster presentation competition, and what is it that he as the judge of this poster presentation competition is expecting from the students, from the posters, and what according to him is the best way to present a poster so that the participants can walk away with the exciting prizes. So without any further ado, it gives me immense pleasure to welcome to the stage today Mr. Darko Medin, and I would let you take over from this moment.
Thank you very much, Suprajit. As you said, I will be talking about bioinformatics and data science challenges today, but also sharing some tips for future bioinformatics experts on how to resolve certain problems, how to overcome certain obstacles in, let's say, data science, and be more successful in bioinformatics career. As Suprajit mentioned, I am a data scientist with previous experience in conducting different research. I was former program mentor for Pine Biotech data science programs, and I will also be one of the judges at the upcoming OmicsLogic Research Symposium poster presentation competition. So I will focus on different aspects of challenges that may arise during bioinformatics research and applying data science to overcome these challenges. So this is the main focus of today's session. Before I actually start addressing different challenges, I must think about where these challenges arise from. What is bioinformatics? What are the domains where bioinformatics is applied? So, some of bioinformatics challenges are actually related, as we can see from the name bioinformatics, biology. So we have the bio prefix in word bioinformatics. It's clear that biology is one of the topics that is of interest, but not just biology. Bioinformatics is a very diverse set of uh, interdisciplinary programs or areas of science, uh, which is related not just to biology, but also to other sciences like chemistry or physics, biomedicine, and clinical research. So when I say bio in bioinformatics, I don't mean just about biology. There are many other sciences which are related to bioinformatics. What is in common for all these sciences is that they are somehow related to a life science. So we can say that bioinformatics is an interdisciplinary program or interdisciplinary set of research methods which are related to a very diverse spectrum of life sciences. So this brings me to challenge number one, which is knowing the domains of life sciences where bioinformatics is applied. I will give a few examples. Suprajit mentioned some of them earlier, like oncology. Oncology is related to molecular biology, it's related to biomedicine, it's related to clinical research too, it's related to genetics. So a lot of different domains are connected to this one aspect of bioinformatics. There are many other areas of bioinformatics like evolution, like plant biology, like studying different molecules, studying, for example, proteins and what happens with the chemistry and physics of proteins. So identifying these challenges is very important in terms of understanding where these challenges lie in which domains of life science. Of course, having domain knowledge is one of the essential parts of any research project, and the research project should have a detailed description of these domains. On the other hand, these domains are important because they're also included in interpretation of results, in understanding what these results mean. So bioinformatics results are not just uh, some numbers or graphs. They are related to real life science problems and ways to address these problems. Okay, so to address these problems, to resolve these life science challenges, which bioinformatics is used for, there is a diverse set of methods and tools which is utilized to achieve better results and to resolve some of these challenges. Some of them are genetic algorithms. This automatically tells me that genetics is one of the main domains in bioinformatics. Machine learning is another set of tools and methods used to resolve various bioinformatics challenges. Data science, as mentioned before, is of course related to both of these areas and is another set of methods and different approaches to resolve bioinformatic challenges. Sequence alignments, logically, if we are talking about genetics, we're talking about sequences of DNA. So sequence alignment algorithms and methods is another set of tools used in bioinformatics. And in the end, but not less important, statistics. Statistics is a set of methods used to analyze and interpret results uh, that are achieved during the research project. One thing which is very important is that most of these methods are applied to a certain type of data in bioinformatics. And this type of data is mostly originating from molecular biology. So I can say it's mostly DNA, RNA, proteins, different sequences from other molecules. So molecular level of biology. On the other hand, it's not limited to molecular biology. There are many other biological types of data which are included in bioinformatics. So as I said before, it's a very wide spectrum of different domains and methods that are applied in bioinformatics. So one of the main challenges, of course, is understanding this variety of methods and challenges. One thing that is in common for all these methods is that they rely on data. So these methods are actually related to data and computer science. So we can say that it's a data-driven set of tools and methods where computer science enables to have different solutions for all the challenges that I spoke about before from those life science domains. So challenge number two, of course, is related to data. As you can see, all the methods are related to data. So understanding how these methods could be applied to different research projects, of course, requires to focus on the data and understand the data that is included in the project. As I said, most of this data comes from molecular biology, so understanding the data, of course, requires understanding molecular biology to a certain extent. So challenge number two would be challenge related to understanding the data and focusing on the data. On the other hand, understanding these tools, these algorithms, and how to utilize them on different types of data, how to get the best results, and by doing that, how to resolve those challenges. So here is an explanation of one very important area of molecular biology, how information is transferred from DNA, from genes, to proteins, which are biologically active substances. So there is a certain flow of information from genes. There is a process called transcription, where these sequences are read and information passed from DNA to messenger RNA, and then at the end, proteins are created by certain type of coding where three nucleotides code different amino acids. So this was just one example of how understanding certain domain of molecular biology is important, because a lot of data in molecular biology and bioinformatics is actually DNA sequences, RNA sequences, proteins, etc. So why would we need to understand these domains of molecular biology? Well, here is an example of how DNA variation is very important in understanding cancer, or understanding how cancer develops, how it's treated, and what are the potentials in developing future therapies, and understanding the principles of cancer development. So here's just an explanation of how mutations, for example, can alter the functions of proteins which may lead to different changes in cells and DNA, which are related to cancer. So understanding these changes in genetic information is essential in cancer bioinformatics. Understanding types of mutations, how they affect the proteins, is one of the examples of how knowing the domain in which research is done is essential in conducting the actual bioinformatics project. Of course, these molecular biology segments should be defined in detail in every research projects related to these areas of bioinformatics. So explaining them in a very concise way that anyone can understand. On the other hand, explaining them in a way which covers all important areas of a research project is one of the, let's say, tips that I can share for this part of bioinformatics. To understand what bioinformatics challenges are, I will first need to understand and explain some similar areas of life sciences and computer science. One such area is called computational biology. So as it can be seen, there is life science domain knowledge, as I previously explained. There are some scientific goals associated with this life science domain knowledge, but this time there are theoretical models and computational simulations. So computational biology is mostly focused on simulations and theoretical models. On the other hand, bioinformatics is focused on real data originating from life science experiments, from laboratories, and statistics and data science are applied to analyze this data, to derive insights from this data. One thing that is in common for these boxes that I painted in yellow here, so life science, real data, statistics and data science, they are both applied sciences. So they are sciences used to resolve challenges originating from the real world, from real experiments, and as such, having data originating from them as real data. Statistics, on the other hand, is part of science which was developed by mathematicians that wanted to apply mathematics on real life challenges. So it's another form of applied science. And that is one of the aspects of bioinformatics. It is, of course, very practical. Data derived from experiments is real laboratory data. And this brings me to another challenge, which is to understand the practical aspects of working with bioinformatics challenges. One of the best ways to learn these aspects is, as was mentioned previously in one of the programs, Pine Biotech by Mr. Ilya Brodsky, is learn by doing. That is one of the best ways to learn a certain method, to understand this method, to understand the whole project, is to actually perform the project. So learn by doing would be another tip that I can mention in this moment. To continue with the whole story of data-driven science. So bioinformatics is data-driven science. I will first go back to some moments in history. So here what is presented is a number of researchers that uh, actually discovered the structure of DNA in 1950s. And of course, from 1990s, uh, Human Genome Project actually improved many areas of bioinformatics. The amount of data was increasing gradually with development of different types of laboratory experiments, different types of instruments able to sequence genes, proteins, etc., etc. What is very interesting is that as time moves on, in past few decades, there is a very strong increase in the number of sequenced genomes, in the amount of bioinformatic data available. So as it can be seen from 2015, there is a very high increase in the amount of bioinformatic data available. Why is this important? I previously said that bioinformatics is data-driven science. If it's data-driven, that means that when there is more data, if the amount of data is increasing, that means that possibilities and development of bioinformatics is also improved. When I say data has increased to such a level, to such an amount, that there is an actual term to describe these large amounts of data that are present today. It's called big data. Now, big data and large amounts of data, of course, have different challenges that accompany them. So with development of big data, also different challenges arise. And a data scientist or a bioinformatician needs to have a different set of solutions, which are mostly IT solutions, to resolve these challenges. So one challenge is data comes from different sources. Huge amounts of data are created all around the world at different labs. They're stored in different repositories. So different sources mean that they will definitely have different characteristics. So one of the challenges is how to resolve this big difference, how to address this big difference that exists within these sources and between them, of course. Another aspect is noise. Large amounts of data produce large amounts of noise. So this is another challenge that needs to be addressed. Large storage requirements. If large amount of data is created, it has to be stored somewhere. This storage requires hardware, disk space, cloud space, different solutions where this data can be processed, stored, and then accessed when needed. Computation power. To analyze huge amounts of data using very complex algorithms that I mentioned before, like machine learning, computation power needs to be there to support these analyses. So computation power is one of the challenges. As the amount of data increases and different types of complex methods are applied to this large amount of data, computation power must also increase. In the end, structure. Structure means that there must be a specific way this data is stored so that future researchers can easily access it. There must be a set of rules this data is stored by, and we can call that a structure. Okay, I mentioned repositories before. So these are the places where data is stored. These places also must follow the increased demand for storing large amounts of data. Over the past few years, a lot of these repositories where data is stored have become open to all researchers around the world to utilize them in different research projects. So I think understanding and knowing how to navigate these platforms is very important. Some of them uh, are mentioned here, like NCBI, TCGA, MBL, DDBJ, UCI, etc., etc. I will explain some of these briefly. So NCBI is a US repository for storing the large amounts of bioinformatic data. TCGA is also used in similar fashion. It's mostly focused on tumors. EMBL, European Molecular Biology Repository, is also used to store large amounts of bioinformatic data, and open. Uh, it's open in large quantities to researchers around the world. DDBJ is another repository from Japan, UCI Machine Learning Repository, etc., etc. So one of the challenges in data science is acquiring data, right? So having open database repositories makes that challenge much easier to resolve. So that's another tip from me to know how to use these open repositories, how to navigate these platforms, how to access the data. So that's pretty much it for this part on data repositories. Now, to understand the data, I said before that focus should be on the data. We first need to understand where this data comes from. Okay, it comes from life science domains, but what are the instruments used? What are the labs used to create this data? So here you can see on the right side one sequencer, which is used to actually create sequence data from different molecular biology samples. But there is another aspect. So definitely understanding the instruments where data comes from is important. And generally, every larger project, even the projects that don't belong to large studies, should generally have detailed explanation about what instruments data came from, what are the characteristics of these instruments, maybe what are limitations of the instruments, etc., etc. But there is another technical aspect that is not that much discussed in bioinformatics communities. I think it should be discussed much more, which is understanding the computer hardware. Why? Well, for a bioinformatician, computer is his lab. That's the tool that his or her lab, as I said, that's the tool that is used by most bioinformaticians to resolve certain bioinformatics challenges. So focusing just on other aspects without understanding the very tool used for direct bioinformatics analysis, which is, of course, a laptop or a computer or a supercomputer. Some labs have special supercomputers to use for bioinformatics challenges. It's essential to understand the hardware needs for a computer. Let me give an example. Analyzing large amounts of data maybe requires a processor that can support this analysis. Sometimes, to speed up bioinformatic analysis, data scientists use parts of uh, computers which are called graphic cards or GPUs to speed up the process. Then, large data sets sometimes require large amounts of storage space on a computer. So understanding all these aspects is critical. So another tip that I would share is it's of course important to know all the aspects of life science domains, to understand the tools, the methods, but it's also important to understand the computer components needed to perform a certain analysis. Okay, learning about sequencing instruments, as I said before, is important because they're generally part of the research project. So to understand the quality of the data, we need to understand the instruments that derive this data, where this data is created. Okay, now I talked about hardware parts. Now let me transfer to one aspect which is not related to actual hardware, to instruments and computers, which is the software part, or how is this data stored in computers, in which formats. Here we have we can see different formats presented like Fastq, FASTA, GTF, SAM, BAM. These are all file formats. I will just make a parallel here for everyone to be easier to understand. We have different file formats on computer, like Word formats, Excel files, images, bitmap images, for example. These are all file formats. In bioinformatics, there are also different formats that need to be understood for practical application of a research experiment, analysis, and then, of course, finishing the research project. It's not very frequent that these file formats are included in research projects, but to be able to practically conduct the analysis, it's essential to understand these formats. What is Fastq format? What is FASTA? For example, I'm just going to give a very short explanation. Fastq files are used to store sequences of, let's say, RNA, but not just the sequences, also the amounts of expression for different RNA sequences. On the other hand, FASTA file is used just to store sequences, right? There is no expression, there is just sequence information. So understanding these very, very specific differences means that researcher understands the actual data that is stored in those files. Another example is if someone is to perform, let's say, RNA expression analysis, he or she should use a Fastq file. But if a bioinformatics researcher is performing just sequence analysis, for example, mutations, FASTA files are one of the choices. There are other files like VCF files, etc., which are similar, that could be used. So I will just give another tip, which is it's important to understand the file formats. That's another important aspect. Okay, so here's one of those formats that I mentioned before, which is the VCF file. So it has a certain metadata, which is not related to these parts of the data essential for the analysis, but it's important to know this metadata often contains information about what type of the data is used in the research, sometimes about instruments, about experimental conditions. So it's important not to forget information contained in the metadata. Okay, so using different formats is essential to understand. Now, next step, once the data is understood in detail, and this, let me call it data understanding challenge, is resolved, next step is to actually analyze this data. To analyze the data, different set of methods are applied. In fact, most bioinformatics analyses are performed in a way that multiple of these methods are used on different types of data. So they form something which is called a bioinformatics pipeline. This is an example of using server T Bioinfo, which is a Pine Biotech platform for creating these pipelines. This is an example of how uploading the data and then using certain algorithm called Bautai, and then using GTF processing, visualization, is used to actually visualize first map the data on the genome and then visualize it using different algorithms. So this is essential for another area of the bioinformatics, which is understanding the pipelines. How can these pipelines be constructed? All of these methods must be compatible one with another, and they also must be compatible with the type of the data which is used. So this is another tip I would share, which is define the analysis pipeline in a way that functions of these methods are compatible with each other. Okay, so here's another example, which is mutational analysis pipeline. I mentioned before that mutations are, for example, very important in cancer research. So here is a pipeline that uses PCR thing, Bautai, Strelka, etc., to actually create a certain analysis which is related to discovering mutations or variants, in this case, in the data. Okay, so once the pipelines are created, once they are defined, and I will just repeat this again, pipeline of these methods is another important part to define in any research project related to bioinformatics. Once that is finished, there is another step which is called interpretation. So as you can see here, there is a graph with different colors and different hierarchies, different lines. There are a lot of details in these graphs. So how to make this result easily readable to any audience, to researchers who are not bioinformaticians, who don't understand all of these concepts that we were talking about, is another challenge. So interpreting the results should be a process where the results are presented in a way for anyone to easily read, to be understandable to a wide spectrum of audience. So I'll just give an example. The left graph has two very distinct groups, one which is colored in red. So we can see that there is a number of these samples which are very high in expression of certain gene. And then there is another blue group here, which has a lower expression of certain genes. So using a very simple way to explain to anyone what a certain result tells us is one very important challenge in bioinformatics. So, as I said before, there are many ways of presenting results. The key is presenting them in such a way that is easily understandable. Okay, so as I said before, some of the tips are to actually understand how to explain the noise, how to explain some solutions to that noise which is present in large data sets, and how to correct certain biases which may be contained in the data. Okay, so this brings me to a challenge which is more of importance for people who are interested in pursuing bioinformatics or data science career, which is knowing uh, different programming languages. Now, of course, these pipelines that I shown before are meant to facilitate bioinformatics analysis without actually knowing how to program in different programming languages. But knowing these languages is a plus because it allows scientists to actually customize the analysis, customize these outputs, visualizations, etc. It's best to know both. It's best to understand how to create different pipelines, let's say on a platform like Bioinfo, but also understand these programming languages to be able to customize the analysis, to customize some outputs, etc. Okay, so R and Python, which can be seen here, are some of the programming. They're actually the most popular programming languages used in bioinformatics analysis. So these are some of my recommendations for any future data scientists or bioinformaticians to actually try to understand these languages as much as possible. Now, with development of large amounts of repositories and data, there is also development of novel methods that are very advanced and can efficiently be used to resolve different bioinformatics challenges. One of these methods, for example, is called deep learning, right? So deep learning is a set of methods that uses artificial intelligence in a way that different algorithms are being used in a similar fashion to that of a human brain to actually learn from the data and learn different patterns to be able to predict different outcomes, etc. So there are different platforms like TensorFlow, PyTorch, etc. I will mention these as very important, but as I said, this is something for future data scientists to actually keep in mind. It's also used in bioinformatics research very extensively in the past few years. So that's one of the areas I can recommend for future bioinformaticians. Now, some key concepts that I would actually mention from this last part, which is interpreting data, is simplify the complexity when interpreting and teach a data story. Being a good data interpreter is like being a good data storyteller. So you must actually teach this story to someone who hasn't been included in the research, to teach it in a way that is easily understandable to anyone reading about the research project. Okay. I know that we don't really have too much time, so I'll just leave this last part of the session for Sukrajit for questions related to any other areas.
Thank you so much, Darko. That was an amazing presentation today, amazing session. I'm sure all of us learned a lot of things about the different aspects of bioinformatics, data science, and next generation sequencing technologies, and also the various methods and ways to address all the challenges and difficulties which we might face in our day to day projects. So, on behalf of the entire team of Psionics, I would really like to thank you for your time and your great presentation. It has been a pleasure to have you here with us today. Thank you so much, Darko.
You're welcome, and it has been really a pleasure for me to conduct this workshop. And thank you very much for inviting me as a guest speaker. So I hope that the workshop has helped address many challenges. And please let me know if there are any questions from students or any other aspects that should be discussed.
Thank you so much, Darko. So, to our audience today, if you have any questions regarding bioinformatics, data science, whatever our guest spoke about today, or if you have any questions regarding poster presentation, the symposium, and other training opportunities and courses, please feel free to put your questions in the chat section. And I will definitely take up all those questions and would try my best to answer you. And while you are typing your questions, thinking about it, I would like to remind you about some exciting opportunities and possibilities in the near future. So, in the month of March, we at Pine Biotech and OmicsLogic have organized several workshops and webinars for you to join and benefit to learn about different popular terms, cutting edge technologies in today's generation. So, as you can see on your screen, we have a very interesting workshop coming up on March 15th to 17th, which is on machine learning and omics data analysis, which is something that is like really related to what we were talking today, what we learned from Darko today. So applying machine learning for doing this big data omics analysis is something which we will be covering on March 15th to 17th. Then we have a special faculty and mentor workshop where we will be talking about several specializations, which includes precision medicine, infectious diseases, neuroscience, oncology, space omics, agriculture, AI and drug discovery, and what sort of approach we take at OmicsLogic, that is the project-based training modules, will be discussed in this workshop. So you can also attend it to get a flavor of what we are trying to do and achieve. Then on March 22, we will be having a detailed analysis of cancer mutations using next generation sequencing workshop where you can learn about the various algorithms, the various pipelines, and the various tools and techniques that we use here to provide a detailed analysis of cancer biology and analyze the next generation sequencing data. Finally, all of these workshops and efforts lead up to the mega event of this month. That is the Omics Research Symposium that is going to happen on March 31st and April 1st, 2022. You can find the registration links in the chat. It's completely free to be a listener, to join and listen to all the talks by the great speakers and the panelists lined up. You can find the link to the schedule as well in the chat so that you can get an idea of the timing. All timings are provided in Indian Standard Time, Greenwich Mean Time, and Central Daylight Savings Time, so you can adjust according to your time zone. And also, if you're interested in submitting your project as a poster presentation, you can find the call for abstract link in the chat. And the deadline for submitting your abstract is March 20. It is an extended deadline. The previous deadline was March 10, but due to the overwhelming response, the great amount of submissions, and requests from the students, we have decided to extend it for 10 more days so that all of you can participate in it. I will be talking more about it later, but We have one question in the chat section, so I would pick it up. The question is from Rizzi Makoka, and the question is, would it require one to understand all the intertwined aspects in order to better answer biological question, or would it require the use of a multidisciplinary approach in a team setup to solve this? So, Darko, if you would like to answer this question, basically, how much or what are the various different types of techniques and skills required to answer a biological question? Is it necessary for one individual to be expertised in different factors or sectors, or it is better to work in a multidisciplinary team where different people are expertised in different segments? So, what do you think, Darko?
Thank you very much to Prajit, and thank you very much, Rishi, for the question. It's a very interesting question, and it's present not just in bioinformatics, but also in data science, the part of science where a lot of research projects that I have performed previously were in question. So, a very interesting question. Bioinformatician, as we have seen from the presentation, bioinformatics and its challenges are a very wide spectrum of different types of science. So biology, biomedicine, data science, machine learning, computer science, statistics. Very often, these analyses are conducted in multidisciplinary teams. So there are computer scientists there, data scientists, there are biologists, medical doctors, etc. But for a bioinformatician, it's important to know all these aspects to a certain level. Now, no one can really know these multi-dimensional approaches to to a level where every single detail is covered, but there is a level where it's important to understand all these aspects, because bioinformaticians often serve as a bridge between, bridge in a symbolic way, between different branches of science like biology, medical doctors, computer scientists, software engineers, etc. So definitely the question is great, and some summary of this answer would be: it is important to know all these domains to a certain level, to a level where a bioinformatician may serve as a certain communication bridge between all these sciences.
Thank you so much, Darko. That's a great explanation and a great answer to the question, and I hope that Rizit really answers your question. Thank you so much, Darko, for sharing your thoughts and answering this question. So, moving forward, this is all about the Omics Research Symposium. Here's the QR code to register for the symposium. We are just a few days away from this mega event. So if you haven't registered already, please scan this QR code. You can also find the links in the chat to register for the Omics Research Symposium, and we would be really grateful to see you at the symposium. So as I was also mentioning about the poster presentations for students and researchers, the four categories, once again to reiterate, are omics and bioinformatics, artificial intelligence, machine learning and data science projects, astrobiology and space omics, and computational biology in the biotechnology and pharmaceuticals. There are several exciting prizes to be won. The deadline was March 10th, but it has been extended on popular request, and the new deadline is March 20. That is a 10-day extension for submission of your abstracts. Here are the rules for submitting your abstracts. Make sure that the length is 150 to 200 words, and it should consist of the following segments like objective, methods, results, and conclusion. The font size should be 12, and the font style should be Monsterrat. And please convert it into a PDF file and then upload on our server. So when you scan this particular QR code, you will be redirected to our website where you can get the form. So fill it up and upload your abstract, and we will analyze and get back to you through email very, very soon. So Pine Biotech has a global presence, and you can find us all on almost every social media platform. So it is a very good idea to stay in touch with us because we constantly keep sharing a lot of information, a lot of updates, and various insights. So you can find all the links to the various social media handles on the chat. So make sure to follow us on all the social media handles and also join our various groups. We have groups on Facebook, on LinkedIn. So in these groups, we have various experts and students from all over the world. So you can interact with them, communicate with them, network with them. So please make sure to do that as well. And finally, you can also share your feedback with us by scanning this QR code and share your insights with us about how you felt in this webinar. What are the various areas that you believe in which we can improve? And all your comments and views will be really appreciated. That's the way which motivates us to keep doing this every week. So that brings us to the end of today's great Psiomics webinar. It was amazing to have all of you with us today. Once again, on behalf of the entire team of Psiomics, we would like to thank our guest, Mr. Darko Medin. It was a very insightful and we learned a lot from your talk today, and we hope to keep learning from you in the days to come. Also, thank you to everyone for joining us today in this journey. We will again see you in the next week with another exciting topic, another exciting theme, with another great speaker and a great guest. Till then, make sure to stay in touch with us through all the channels that we have. Feel free to send your email to me if you want. My email ID is communications@pine.bio. I'm putting it down in the chat section. You can send me an email if you want to communicate anything with me. I will be more than happy to answer you. So if you have any questions, feel free to put it down in the chat. Otherwise, that's a curtain call from us today, and we will see you again soon. Take care and stay safe. Thank you so much for everyone for joining, and we will see you soon.
Thank you, Suprajit. Thank you, everyone, and best of luck in the symposium and competition to everyone.
Thank you so much, Darko, for your support.
Thank you.